This Wednesday morning, January 30th, 1991, we are at Becton Dickinson Immunocytometry Systems Company in San Jose, California, in the office of its president, Dr. Nagesh Mhatre. Dr. Mhatre, the objective of our project is to document the history of the cell sorter. But first, let's talk about the company that was the first commercial manufacturer of this instrument. Tell us about the history of Becton Dickinson and Company. Where and when did it start, and what does it do?
Becton Dickinson and Company started some 95 years ago. Two salesmen from North Carolina met in New Orleans on their trip to San Francisco, became good friends, traveled together. When they returned to North Carolina, they decided to go to form a company that will be for medical products. And they started their first factory, so to say, in New York City. At that time, they used to import glass barrels to make the syringes, and later on they started to make thermometers also. These syringes were imported from France and Germany. And when the World War I started, they had to begin to make their own syringes, barrels, glass barrels to make these syringes. And so they started a factory in New Jersey. So initially it was using these glass barrel, making syringes, hypodermic syringes, glass syringes, and later on thermometers and other medical products they started to make. Over the next several years, the company started to grow and played an important role in World War II to provide the Army the necessary medical products. In early 1950s, the company started to acquire small companies to get into the diagnostic field. As a result of that, we have a very large diagnostic sector today. And in medical products, we have continued to add many more products, catheters, thermometers, then acquisition of another company, Deseret, added some more acute care products. On the diagnostic side, we have today the whole area of microbiology, that is prepared plate media. We have instrumented microbiology products, instruments. And in Vacutainer, which is another large division, we has blood collection systems. Then we have labware products, which makes pipettes and plates, which are used by scientists, and tissue culture products. And then we have primary care products, which are used in the doctor's office, like QBC and IQ. We also have a malaria test now for tropical medicine, and this is just being launched recently. With that, we also have an immunochemistry product line that is one new introduced called Affinity, which is also very recently introduced. So we have a very major diagnostic business. About 60% of our business is still medical and 40% is in diagnostic. In addition to all these divisions I described to you in the diagnostic sector, the history of Becton Dickinson immunocytometry should be added. In 1972, for the first time, when Bernie Shoor, who was then general manager for BD Electronics Laboratory in Mountain View, in collaboration with Stanford University professor Hersenberg, got the idea of making a cell sorter. So they combined their talents and produced the first commercially available cell sorter, and that was in 1975. And they named that product FACS, acronym for fluorescent activated cell sorter. And we formed that as a division in a commercial sense in 79. And it was again in 1977 when Dr. Milstein published the paper on hybridoma. Again, Hersenberg and Bernie Shoor got together and started a small organization here called Becton Dickinson Monoclonal Center, Inc. And that also started to grow. And both these entities were separate, and they were run separately by the corporation. It was in 1983, the chairman of the company asked me to come to California from, I was in Europe at that time, and take over Monoclonal Center. And in September, they asked me also to take over the fax organization. So we combined these two organizations in January of '84 and came up with the name Becton Dickinson Immunocytometry System. So this is the beginning of what we had. Originally two separate entities combined for strategic reasons, and that proved to be a very good decision.
Give us a sense of scale and of the growth. How many people and how much money was involved in the early years, and how has it grown since?
Let's take the fax division. As I mentioned, it was only few people who were there in 1975. But by 1983, in fax division, we had something like 110 people involved. Practically most of them were engineers at that time. Whereas the Monoclonal Center, which was started in '79, had about 63 people in 1983. So you combine these two units, we had about 180 people. And that was in 1983 December. And today we are looking at some 700 people for BDIS in the United States. And then to that you add another 300 people overseas. A large chunk of that comes from Europe. And on the scale of revenues, in 1984, the BDI's combined revenue was somewhere about $15 million. And now in 1990, we're looking at upwards of $150 million. So that gives you the kind of growth we had, and a whole industry is expanding.
Although you are a very successful manager, you're not a manager by training. Give us some idea of your background and early career and what brought you to BDIS.
Well, let me just very briefly give you my own background. I was trained as a scientist. I got my PhD from Rutgers University in biochemistry. And I joined Miles Laboratories, where I was a research scientist. And I remained as a research scientist for the next 10 years after I got my PhD. And I was a senior scientist and director of a laboratory, Ames division, of Miles Laboratories. And I was enjoying doing the basic research at that phase. A problem came with a company that Miles Laboratories had started in Israel with Weizmann Institute. And I was going to go to do a sabbatical at Weizmann Institute on immobilized enzymes. And the chairman and the president of the company called me in, and he says, why don't you go and run a company? It's a joint venture. It's a small company. I will give you good experience. And it's science-based, first biotechnology company in Israel. That was in 1971. So I thought it would be a great challenge, a new kind of work for me, and I had to learn on the job how to run a joint venture, which was 60% owned by Miles and 40% by Weizmann Institute. The company was in the red when I arrived there. We didn't have money to pay the employees there for a monthly paycheck. And when we added new products, new technology, it took off. At the end of 4 years, we were awarded Outstanding Exporter of the Year, and we were able to show profit for the first year in the company. After that, I came back to Miles, vice president of one of the divisions, and the company was bought 2 years later by a buyer, a German company, and I felt much ill at ease to operate in a different environment that was created from what I was used to when the Miles was owned by the American organization. Becton Dickinson called me, and I came for an interview, and I liked their approach, their atmosphere, their culture, and I took the job as a group director for business planning for diagnostics. A year later, I was appointed to the position of president of Becton Dickinson Europe for the diagnostic group. And I spent 3 years. My major task there was to organize Europe as a one business entity rather than 10 countries, and each individual country operated differently. And you have to remember, this is 10 years ago when Becton Dickinson was looking to unify, to make a common European economic community. So all of these things happened rather quickly, and I had to learn on the job marketing, sales, human resource management, along with new technology. So it was just a learning process, and I took courses, read books. I don't have an MBA, or I don't have a formal business training, but I think it's a scientific training that made it possible to learn other things.
And what motivated you to go from an academic setting first into a corporate laboratory and then into management? What sort of motivates you to do that?
Well, one of the things that I think I get really turned on is to find answers to the questions that pop up, and I'm curious about new things that are happening around. But I wanted to use my scientific training to make some tangible products, and I felt that the industry provides that kind of resource to do those studies. And at the end of your research, you actually see some tangible product. As a matter of fact, in the 60s, early 60s, very few people with good background would go to work for industry. My professor felt somewhat dismayed that I chose to go to industry instead of staying and pursuing the academic background, become assistant professor and all that. And but I looked back and I thought that was a very good decision, because I was able to come up with quite a few new products and a few patents which are today on the market. So what really drives me to see something useful, tangible you can produce with your scientific knowledge. And this has continued here as well.
Now, some say that in the academic world, science is open, there's free communication of ideas, whereas in the corporate world or industrial laboratories, that there's many more restrictions, there's more secrecy, or more closed science, as you may. Well, what's your experience since you've been in both worlds?
Well, paradoxically, as I found out that it's the other way around. That is, in the industry, like right here, we have very open communication. There's a lot of teamwork that goes on between departments, different disciplines of science and marketing, finance, other groups. Unlike academia, where it's just one-man show, and they don't even talk to each other. They will not reveal to each other some of their ideas in their own departments. There's a lot of rivalry. And part of the rivalry probably is also that the kind of resources we have to do our work are much more greater in industry than one would find in academia. So my experience has been an atmosphere where there is open communication, there is a lot of teamwork that goes on, and a great deal of synergy is produced, and I think you have a more effective way of getting better results.
You touched on the international nature of BDIS and BD. Could you talk a little bit more about the sort of transnational aspects of the company, and how that affects its management style, the way people interact in the company?
I think, as a matter of fact, one of the major reasons, there are many reasons why we have been successful at Becton Dickinson, and particularly here at the BDIS, Immunocytometry Systems Division, is that we have seen our business as a global business, and therefore we have deployed our resources across the board. For example, BDIS business today, 60% of it is outside of the United States. And we really exploit the talents that are available anywhere in the world. For example, we have people here in this building in San Jose, some people from 26 different countries working. And we also move our people around the globe, literally. For example, one of the fellows who used to work for me, now he's a business director in Europe. He's an American. He runs our total European organization for BDIS. A young lady from here, an American, she's now our product business manager in Japan. And we have a Frenchman who is running our Asia Pacific business, and we have an Italian who runs our diagnostic group in Japan. So we have moved people around, and we are nationality blind. And that really allows us to have a global approach, global thinking, and operate as a transnational company, so we can leverage the kind of money we spend on research here for a much larger market. And fortunately, the business we are in is transnational in the sense the science that is done in China or in Japan or in Korea or in New York, it's the same thing. These are the same scientists. They come to the same scientific meetings. They read the same journals and magazines, as well as written in English.
So you see science as the common language that binds
yes,
you
both
all?
science and medicine, as it is practiced around the world, there is a great deal of commonality, common denominator, brings them together.
But obviously there are cultural differences. And how do you deal with these cultural differences?
Yeah, one has to really, I believe, live in another culture to understand that you don't make these moral judgments about other cultures and adjust to why they do things the way they do, and to be effective. For example, to give a speech in Japan, one talks a great deal with humility and apologizes right from the beginning for being there even. And then you can get your message across. Now, you wouldn't do that normally here in the States. Whereas if you're doing business in some parts in Southeast Asia, you do most of the business in the evening after 5 o'clock outside the office. Whereas you go to Germany, you do business right in the office. You're done with it. You shake hand and you leave. So there are cultural differences, but one has to understand how to respond to these different environments we work with. We have here in the States as well. If you think about it, on the way you do business in New York City compared to someplace in Alabama, they don't like to be very aggressive approach. So we have a different kind of salesman even within the United States who operate different.
Dr. Mhatre, could you describe your management style and the way the work is organized in your company?
Well, let me make a first statement that it's not my personal management style alone. It's a corporation's culture, and you will see this done in other divisions of Becton Dickinson as well. Basically, what we try to implement is an open communication. We want to know what employees think about the organization and how they can make contribution to improving the way things are done. We do not make unilateral decisions. It's not a hierarchical organization where there's a great deal of teamwork that goes into implementing the decisions that are made. And therefore, we try to give as much credence to what our employees have to say in terms of making impact on the decision that is made. And the decision-making process is driven on the spot where the decision has to be made rather than some office that is remote from where the action is. And that's the basic governing principle. And the results of all of this is, for example, people feel ownership of the company. They feel involved in the company. They bring out the innovation. They give extra hours that are required to be the best in the industry. Our whole emphasis has been on the superior quality, and I think that's what differentiates Becton Dickinson from other companies. Now, superior quality is not only the products that we make. That involves services, how our customers relate to us, the long-term relationship we have developed with our customers. These are the factors that finally are the result of the kind of culture we have developed. And I think we have adopted some methods in manufacturing, for example, where there is a great deal of emphasis on quality rather than the quantity of products we put out. And as a result of this, when the product is finished, it is 100% okay to be shipped. So we don't make 15 products and then 3-6 of them are chucked out because the quality isn't right. We make, for the first time, whatever is made has to be done well. And that goes not only in manufacturing instruments, reagents, and so forth, everything that we do. We like to think that we do it for the first time things right. So we've placed a lot of emphasis on training of our people. We have here, BDIs, 2 full-time trainers, and there's a continuous training that goes on. There's a lot of emphasis on learning, understanding new techniques, understanding new management methods, so that we continuously try to improve our performance.
What kind of people do you hire?
Well, we spend a lot more time in screening people before we hire them. Sometimes it seems very tedious and long process, but the key to really management is to be able to hire very, very well-qualified, high-quality individuals who have a high degree of integrity, personal integrity, and have an attitude to do better every day. And so we go one step more to hire really good quality people here.
How hard or easy is it to get these kind of people?
Well, I don't know whether it's very hard or easier, but because of the reputation we have established here, and our own employees are pretty good salespeople for this reason, we have been able to hire and retain very high quality people across the board, not just scientists and engineers, secretaries, support system, in finance and buildings and maintenance, you name it. You will notice that the people who work here I like to high quality.
Is there a private life that you have outside of this
You're asking
company?
me personally?
Yes.
Not really. I think if anybody who has the responsibility I have has to give everything he has or she has for this task. And it is a multifaceted job. You have to deal with people, you have to deal with the financial situation, you have to deal with the customers, and sometimes the Wall Street analysts. All of this is a lot of fun, actually. And a number of people are depending on you to do the best you can. And that leaves very little time, especially when you're involved in the global business. I spend more than 50% of my time visiting our customers, our employees outside of the United States even. So that takes a lot of time.
How do you keep yourself intellectually stimulated? Because you're a man who likes to seek knowledge, and during travels and management, how do you find intellectual stimulation?
I think I don't have to seek it. It's all around us here. For example, I sign up for 3 or 4 courses a year of brand new subjects, computers, new lasers, as well as some management techniques, some things like how do you negotiate better. I took a course at MIT for 10 days because you can improve your skills in all these areas. So there is plenty to learn.
Okay, I think on that. No, we'll see.
Owen, hold for Steve. And Steve?
Good. Just tell Ray.
Welcome to Becton Dickinson Immunocytometry System. We are engaged in a business that produces products for medical scientists and the clinicians, instruments, reagents, and software. And primarily these are today used in the field of immune deficiency disease, AIDS, and cancer. And we have other areas of medicine we are probing to develop new products for the scientists and systems for the clinicians. We here operate on a basis that people who join us are going to contribute in many ways. And we have an open management system. We want people to participate and also tell us what they think about it, have a high integrity, and finally we want to offer total high quality to our customers.
Good, I'll take the job.
Hold for speed. And speed. Go ahead.
Becton Dickinson immunocytometry system is really a part of a larger corporation, and this is a major advantage we have. And Becton Dickinson and Company this year had a revenue of $2 billion. Has two sectors. One is medical sector, and the other one is the diagnostic sector. And we have a very unique relationship with our corporation in that this organization is treated differently in terms of reporting directly to the CEO, and he personally takes interest. He comes every 3 months to spend time with us for a couple of days. And as the corporation is looking at this division as a window on future for the company. And so we have a best of the both worlds, the entrepreneurial atmosphere that we create here, as well as the tie-in with a very large corporation.
Great. Okay. Up to. Rolling. Hold for speed. And speed. Okay, too good.
We are now in one of the conference rooms at the Becton Dickinson Immunocytometry Systems Company, talking with Dr. Bernie Shoor.
Excuse me, no doctor.
A doctor, Mr. Bernie Shoor,
Mr. Bernie.
who has retired from this company about 6 or 7 years ago, but before retiring, he had worked here for about 35 years, and among his titles, he was vice president of the company. Bernie, since you were here from the creation, as they say, or maybe you were the creator, tell us how BDIS got its start.
Well, I was running in the early 70s a small satellite laboratory for Becton Dickinson. This was an advanced development laboratory that took on state-of-the-art developments for various BD divisions. We were making semiconductor sensors for a division in Southern California, and we were working on an advanced physician's office cell counter for the BD Clay Adams division. We became aware of some activities that were going on at Stanford, which involved a prototypical cell sorter in the department of Leonard Herzenberg. And we made contact with Len Herzenberg and his people and found that what they were doing was it was very interesting for us because they were building a machine that could sort cells at very high rates of speed. And it was an engineer's dream in a way. It involved lasers, it involved electronics, it involved computers, it involved fluidics, and it was a pretty fun project. We actually, as I recall, hired some of the people there, perhaps Dr. Hartzenberg, as consultants, because we felt that some of the things they were doing might be applicable to our project. And the chemistry of the relationship worked very well. We got along very well with them. Subsequently, Len approached me and said that he had funding to build 2 machines, which were derivative from the room full of equipment he had in the basement. And these were to be machines, one of which was going to go to NIH, one of which, the other was going to go to Stanford. And they were to be machines that people could use. In other words, you didn't have to be an engineer to use them. I was able to get approval to take on this project. And we reached an agreement with Stanford whereby we would build the 2 machines. I think they were going to cost something like $180,000. And that we also negotiated a license so that if anything arose from that project, any future products, we would have a license under their patents and their know-how. It was hard to predict that it would go anywhere, but it had a basic, what they were doing had to be right, it had to be important at some point, because they were sorting the cells of the immune system, and while you couldn't predict how important they would be, in other words, what sort of diagnostic implications it would have, the fact that there were these cells there and there were different cells with different functional roles in the body, you had to believe that it was going to be important. And so we went on and we built those 2 machines under the contract. As part of the program, we also came up with a complete set of drawings. We came up with instruction manuals. And because it was important that those machines, when they were delivered by us, be maintainable and supportable. We delivered the 2 machines. I think it took a year plus. The work was done by a group of 4 people. And you might say it was a small task force-oriented group. We didn't believe in putting armies on projects like this. We put just 4 people. And the machines worked.
Do you remember the year that these machines were built?
Oh, I think they were probably delivered in late 72 or early 73. The machines worked, and they worked very well. And as a result, other people started to want to buy the machines as well. We had requests from England, from Germany, from other laboratories in the United States. And we took the decision that we would supply those machines. In some respects, it was a rather controversial decision because we didn't have the resources to support them in the field. We couldn't send out service agents. And so we adopted a strategy that said that we would, wherever we could buy a high-quality subsystem, we would buy it, whether it came from Tektronix or Hewlett-Packard or whatever. And the major portion of the machine that we built was the optical bench, which included a laser from spectrophysics and some of the electronics. And when we contracted with the suppliers of the subsystems to the end that they would support those subsystems in the field. Well, the spectrophysics would warranty the laser and go out and fix it if anything went wrong with it. Hewlett-Packard would fix their parts of it. Tektronix would fix their parts of it. So we felt reasonably confident that, we had a supportable system in large measure. And we gave the customers a big fat book of drawings and prints and our blessing. And, we said, you can, this is what we can do. If you want to buy it under these terms, you buy it. If you don't want to buy it, that's fine also. And we were fortunate that this was not our bread and butter project. This was an additional project to the activities of the lab. And so we weren't pressured to sell 5 machines a month or 8 machines a month. We could be very relaxed about how we sold those machines. And I think perhaps this 3rd machine went to London, University College. Maybe the 4th machine went to Cologne at the Institute for Genetics. And people started to publish papers based on the work coming out of the machine. And that really was the crucial element in the machine being accepted and being successful. But the other thing was, as I say, is that we didn't overpromise what we could provide. And we actually, the other thing that made our life simpler is that we sold it on the basis that the customer had to give us 20% with the order in cash. And he had to pay us for the machine on delivery, the rest of the machine. And if they didn't want to do that, we didn't sell it. So we were able to fund our activities from that flow of monies. The thing that happened, and the division grew. We started to sell, 10 machines, 12 machines, 15 machines. And of course, what happened in 1975 was the Kohler and Milstein coming up with monoclonal antibodies. And that did nothing but increase the utility of the machines, the instruments. Because now the scientists had much more specific ways of labeling the different subsets of cells that were in the body. And I guess, again, we got into the monoclonal business at the instigation of Len Herzenberg, who actually called me up from London and said, you've got to go make monoclonal antibodies. And my response that night, I think, was, what are they? We learned rather quickly what they were, and the corporation supported setting up a laboratory to get involved with monoclonal antibodies. The laboratory was run by Chuck Metzler, who we hired out of Yale, who had been a student of Len Herzenberg. That was a major step forward in the evolution of this instrument. I would say if there was another major step that impacted the growth of this field, it was in 1980 was the development of understanding of AIDS and its presence in society, because that gave an enormous impetus to cellular immunology activities. Those were, I think, the 2 major key elements.
Talk a little bit of
Owen, stand by for speed. And speed.
Bernie, could you talk a little bit about the relationship between the engineers that were working at BDIS and the scientists? You mentioned the immunologist club.
Uh-huh.
Can you elaborate a little bit more on that?
And the engineers that were working for us and the scientists that were working in the academic community.
Yes.
We positioned ourselves, and I think we were right, that inasmuch as we couldn't predict where this field was going. We couldn't, and there were so many ways that these instruments could be used. that our basic role was to support the scientific community, provide them with instruments that they needed, change them if they needed changing, build a versatile instrument that was a research tool for the scientific community. And conceptually, it was a tool that could be modified. It could be modified by us. It could be modified by the customers, because many of the customers were leading-edge scientists. They had their own engineers. They had their own electronic people.
So it wasn't a closed black box.
So it was not, no, we didn't. We built, contrary to what most people do, we built an open system. In fact, the optical bench was open, and you could get into it and work with it. The electronics were accessible. And again, it was not in a fixed enclosure that had to be where there was no room to expand. We left lots of room to expand. Because the vision here was that we couldn't predict where this was going to go, what it would need next year, and we had to be flexible. And in fact, subsequently other people adopted that philosophy. And we were, at that time there was formed a society in the middle 70s, early 70s, of people who were working in this field, scientists, and it included scientists in the research community and scientists like our own that were developing machines. And that today is called the International Society for Automated Cytology, and probably has on the order of 1,000 members worldwide. China, Japan, Australia, New Zealand, the Eastern Bloc countries. We have machines in Africa, for example. And many of those people are still part of that organization, which meets every 18 months. The next meeting is in Bergen at the end of August of this year, 1991.
They have a journal as well, correct?
They have a journal called Cytometry. And it's a flourishing, viable community, which trades papers, which trades reagents. And we, in those days, and perhaps I think still today, we really saw ourselves as supporting the scientific community. That was our major goal. And we felt if we did that, as this business sector grew, we would grow with it. And that turned out to be true. Our best friends, our best salesmen, were the scientists in academia. And if somebody came to us and said, hey, why should we buy your machine? What can you do with it? We would typically send them to a university laboratory and say, go talk to those people, go see what they're doing. One of the things that we did is we created a bibliography of papers, reprints, and we sent reprints out to people to show what could be done with these machines. That was our major advertising thrust.
Now, you're not a manager by training.
No.
So tell us a little bit about your career and training and how you got into this business.
How far back do you want me to go?
Back to the Bronx.
Back to the Bronx. All right. Well, I studied physics at New York University, at University Heights, and graduated with a bachelor's degree in physics in 1942, by which time the war had broken out, and there was no option about going on for the schooling. And so I went to work in a government laboratory, actually virtually dragooned into a government laboratory. And I worked there for some time and then went to work for the Sperry Gyroscope Company for a couple of years, primarily in instrumentation. And I guess somewhere in that period I started to run a low-grade rheumatoid type of fever, and I moved to California and worked for some years for a couple of aerospace companies, Douglas and Northrop. Early 50s, I became a consultant because there were some projects that I really was very interested in. And one of them was a bicycle ergometer, which was a machine to measure cardiac output under controlled levels of stress. In 1955, I went to work for a small company in Pasadena that was run by some people I knew that were making aerospace-type measuring devices, piezoelectric accelerometers, pressure pickups, force gauges. And I became the chief engineer, technical director. That was fun. We were building state-of-the-art devices, which is really where I always got my kicks. In 1962, I guess it was, I decided that we really didn't want to live in Pasadena anymore because it was just too smoggy and too unpleasant. And the company took that opportunity to send me to England to operate a small division that we had there. And that, I was really in the role of a fireman, but the fire was too difficult. And we decided to close that. I came back to the United States, and in deference to my desires, I was sent up to Northern California, to Mountain View, to run a small laboratory that we had there. Because I made it clear I wasn't going back to Pasadena, not for any extended period of time. And it was during that laboratory was set up to do state-of-the-art transduction devices originally. Subsequent to that, BD bought the whole company because they had the idea that it might be transformed into a medical electronic company, which was probably unrealistic. The only thing that became transformed into a medical electronic company was my laboratory, and only part of it. And it was as part of those activities that we took on the project for Clay Adams to help develop the cell counter. And that really was the start of our really major entry into medical electronics. We were, because of the success we had with the cell counter out in our laboratory in California, I guess I developed probably an undeserved reputation as a guru. And the company asked me to go back to New York and become corporate vice president of research and development. And because I was perhaps challenged by that, we accepted that.
Was it because of the cell counter or cell sorter?
The cell sorter, excuse me, because of the success of the cell sorter, because it was starting to have an impact. And you must remember at that time, BD was really not an instrument company. It was what I call a glass and plastics company. They made vacuum containers, syringes, petri dishes, scalpels, really the devices used in the hospital, things they used in the hospital. They also had diagnostic kits. They had some position in diagnostics, in radioimmunoassay. But that was not their major thrust. And so I spent 4 years in New Jersey as corporate vice president for research and development. And I guess at that point I felt I'd had enough of New York. New York was a hard place to live as you get older, and if you're a Californian and are used to California. So I told them I want to go back. And I had kept my home here because that was always the intent, to live here. And they asked me what I would like to do, and I said, I think we should build up our monoclonal activities. And so they were supportive. They provided the funding that was necessary to do that. And I returned to do just that. We built new laboratory facilities in the new building. We hired some excellent people. And we started to become a force in making and marketing monoclonal antibodies. I guess a year or two later was when the decision was made to retire. It's partly as a result of a rather unfortunate auto accident in Europe, where I fell asleep at the wheel of a car and drove into the side of a brick building. Not much damage except to my ego. But I decided I didn't have to do that anymore. So we had a general agreement that I would retire and become a consultant to BD. Now, I think the company was reasonably happy about that too, because this was becoming a bigger and bigger organization, and I'm not a paper man in terms of running a large organization. So it worked to everybody's satisfaction. And that's when they brought Mhatre out. And so for the last 7 years or so, I've consulted with BD.
You've witnessed the birth of the biotech industry in Silicon Valley and this area, and that even BD has been involved in some of that. Can you just say a few words about that industry?
The interest, the biotech potential has been clearly there, and a lot of people saw it as a way to make a lot of money. And so in some way it was a symbiosis, if you will, of the venture capital community with the academic people, primarily. And there have been a lot of companies put together based on the ideas, most of which would come from academia. Some of them have done relatively well. Most of them, I think, have not survived. In the best of circumstances, they've been bought out by bigger companies. And then the bigger companies are in a position to pick and choose the things they want to support. There's no question of the truth of the matter that the potential is there. I don't think we've even seen the end of what biotechnology can do for us. And so I think it's been a fortunate situation in many ways.
I think you have a very interesting philosophy of what makes a successful company, a biomedical instrumentation company. Why don't you share a few thoughts on that topic?
Now, which aspect of that are you talking about?
Well, just I think when you were mentioning to me that to be successful, you have to be running ahead of the crowd all the time,
true, yes.
Quickly changing.
A lot of people, the conventional wisdom is that you depend on patents to protect your position. I think in a state-of-the-art business, you can't predict which patents you're going to get. You can't predict which patents, how valuable those patents will be. You can't predict that you're first, and you can't necessarily stop other people from inventing around you. And so philosophically, the only way to go is to run and just, try and maintain a state-of-the-art position. It's not at all a mature industry. And so you really have to get out there and be first in the market. But being first is not enough. You have to have the resources to do it right. And I think that's one of the things which really helped us a great deal, because we did have the resources. We were supported by a major corporation, but the major corporation let us run like a venture. And that's a very unusual situation. Most ventures don't have enough money to do the job properly.
Thank you.
All right.
Pause it for a second.
Rolling, hold for speed.
This particular division really got its start in about 1970, when we were working on a project for the Clay Adams division to make a cell counter, which would be used in the doctor's office. We had a very small group of highly qualified people who were capable of taking on various disciplines in terms of creating new products. We were not going to be the manufacturer of those products. We were developing, showing feasibility and providing the drawings, and then typically we would deliver that to the other division that would then produce it, buy it, and sell it. It was in and we actually made the small doctor's office cell counters. We also made a new state-of-the-art machine called a platelet counter, which actually involves some of the technology that we had learned from Stanford, with a piece of technology that was incorporated in the cell sorter. In roughly 72, I guess, we came to an agreement with Len Herzenberg's lab at Stanford and Stanford University to produce 2 machines under a subcontract to Stanford. These were what later became the FAX-1 cell sorters. The primary contract was from NIH to Stanford. We had the subcontract to build them. And we put a small team of people together, approximately 4 people. And they worked directly with the scientists in Len Herzenberg's lab to essentially take what we would call a kludge, a large assemblage of things all over the tables. We re-engineered it to create a product which we call the FAX-1. The FAX-1 was the first machine the first commercial machine, if I can put it that way, because Herzenberg was doing it before, that could sort live cells by virtue of antibody markers and stains at very high rates of speed. We could sort over 1,000 cells a second and selectively put 2 subsets of cells into separate test tubes. And it was it was very well received, in the scientific community. And we began to get other orders from people around the world, not just the United States.
I didn't interrupt you, just because you're giving more detail than we need. Can you just go very rapidly now to the present? Okay, Bernie.
FAX-1 it was succeeded by FAX-2, by FAX-3, by FAX-4, and instruments that today are called FAX-Star Plus, at the high end of the line. They are distributed worldwide. We have machines in Africa, in China, in Japan, and virtually any country that you can name that has adequate laboratory facilities to utilize them. Any country that you can name that has adequate laboratory facilities to utilize them. Currently, we're building a machine called a FAX-Scan, which uses the same principles as a FAX-Star, but doesn't sort. And that has is getting closer to a clinical instrument. At the present time, we also make monoclonal antibodies, which are sold throughout the world, and which really are complementary to the instrument. So we tell people we're selling a system. We're selling an answer to a problem, which
We are still at the conference room at BDIS. This time we're talking with Dr. Bruce Bach, the Corporate Medical Director. Bruce, could you define the field of flow cytometry so that we will know better how the cell sorting fits into this broader field?
Well, Ray, I think flow cytometry really has two definitions. There's a technical definition of what is meant by the term flow cytometry, and then there's a definition which relates to how the measurements are made. Flow cytometry, as used today, means the measurement of cells in a flow. It is a precise, quantitative measurement of the physical properties of cells. It's accomplished by harnessing the power of laser technology as a coherent light source, optics to focus that light on a target, and to collect the light which that target produces when it has interacted with the laser beam, and then electronic signal processing. These technologies, the hydrodynamics of flow, particle, past a light beam, and the collection of that light, really is the core or heart of this convergent technology.
Now, this technology and some of these have earlier antecedents and roots, and maybe you can describe what some of these are.
Just before you, let me remind you, don't look at the camera.
Okay.
You're talking to Ray.
Okay.
Maybe you can start with the cell and
Excuse me?
cellular pathology and
Sure.
Wait a minute, I don't have to repeat the question, right?
No,
Okay.
fine. Carry on. Okay, I think flow cytometry really represents a development which has its antecedents in the 19th century. With the coming of an understanding that the basis of disease, if you will, the locus of disease in the body was the cell, certainly the work of Virchow and Kontheim and others at the middle of the 19th century, was very important in getting medical people and cell biologists to realize that bodies and organs really were composed of cells, and that the cell was the central functional unit of the body and perhaps the central functional locus of disease. With our ability to look at the microscope, define cells by their pictorial or staining properties, we were able to begin to understand the disease process anatomically and microanatomically. Flow cytometry is an extension of this tradition by permitting now individual cells to be studied functionally and to correlate their physical properties, the display of proteins on their cell surface, their size, their metabolic activity, with their functional activity. So we are making a relationship between structure and function, and doing so in a new way. We're no longer restricted to looking at images which have been put on a single glass slide, but rather we can look at a cell while it's still alive and make multiple measurements of that cell while it is alive, passing in a stream. It's a tremendous enhancement in our ability to understand how cells function and indeed what the life processes actually are and how they're integrated.
Now, you've been involved in this field from early on in your career. Maybe a few words about your background, how you got into the science, and then your experience with the cell sorters.
I actually was very fortunate in terms of my scientific training and career. When I was growing up, I was the beneficiary of the tremendous investments that the United States made in science and scientific education. And even when I was in high school, I was very fascinated in the cell as the fundamental unit of biological organization. My university training was in cell biology and genetics, and that's a theme which I've really followed through and has ultimately caused me to be in the position that I am in now. When I was an undergraduate, we were beginning to understand how the DNA was translated into protein and how proteins actually created a functional program for the cell itself. When I finished my undergraduate training, I went to medical school for 2 years and became very, very fascinated in immunology. Because of that interest in immunology, at a time just when cell sorters were becoming commercially available, I elected to pursue a career in basic scientific investigations of the immune system. It was a very exciting time. When I arrived in Boston, Massachusetts to begin my graduate studies, I was one of the people who helped unpack the first cell sorter, which was established at Harvard Medical School, and we immediately applied this new technology to the understanding of the immune system. Unfortunately, we only had part of the tool set that we needed. We had the ability to make sophisticated physical measurements based on scatter and size. We had the ability to use polyclonal antibodies to mark cells, and we had the ability to conjugate those with at least one fluorochrome, in this case phycoerythrin, no, not phycoerythrin, I'm sorry, fluorescein isothiocyanate. This gave us the ability to make fluorescence measurements of proteins on the cell surface and also to separate the component parts of the immune system into separate pots, if you will. We could use the cell sorter to fractionate the immune system into T cells and B cells and accessory cells. So in the early 70s, when flow cytometry was just being started as a science, we were able to begin the first experiments which allowed T cells to be sorted and then transferred into animals and manifest their functional roles. And B cells as antibody-forming cells could also be studied in isolation. So it was a very exciting period of time. After I finished my graduate studies, I went on to complete my medical studies at Washington University in St. Louis, and we used the cell sorter flow cytometer at that time to study childhood leukemia and the emergence of infectious disease and the immune response to infectious disease. When I then moved on to San Francisco as a house officer, we began to see the first patients which had HIV infection. And in my internal medicine training, we were among the first to apply flow cytometry to the measurement of the T cell disturbances which underlie HIV disease. So in a way, my entire career has been influenced by the availability of the cell sorter and flow cytometers, and in a way I've been a beneficiary and very involved in correlating basic science observations with clinical medicine, going back and forth between the clinic and the laboratory to allow the laboratory tools to understand the underlying pathophysiology of human disease states.
Could you talk a little bit about the people who influenced your career, maybe who you worked with, whose laboratory you worked with at Harvard, and then the teachers at Stanford?
Well, I was very fortunate in that I came to Stanford in the late 1960s and early 1970s, and at that time there was a whole group of young, talented investigators and young junior professors who were really at peak periods of their productivity. People like the Herzenbergs at Stanford who were working in genetics of the immune system, people like Dow Woodward and Charles Yanofsky who were studying the interaction between the genetic code and its expression at the level of proteins on the cell surface. So my basic training was understanding that the functional program of the cell reflected the genetic code, but that not all the genetic code was being indexed, and that in essence we were interested in studying how external forces influenced how the cell responded. So a lot of my early work was based on things like neurospora, which is a mold, looking at the relationship between the cell surface proteins and the genetic code. Then
Were the colonies still from the time of Beadle and Tatum? Or
absolutely, absolutely. My professors had been graduate students with Beadle and Tatum, and one of the important points that was sort of translated at that period of time is that there's a molecular economy, that the entire genetic code of an organism is not expressed at any given moment in time, and that how the cell makes a selection of which elements of the genetic code to manifest in its proteins is an important clue to differentiation and, if you will, the functional properties of a cell, which determine its behavior and, if you will, its life cycle. When I went to medical school, immunology was perhaps the newest clinical science, and it really wasn't defined. I had the opportunity to work with people like Baruch Benacerraf, who later won the Nobel Prize for his work in understanding the immunological system and its genetic regulation. To be able to have access to many of the people around the world who were studying the genetics which underlie the immune response, the genetics which underlie the development of T cells and B cells, all of these studies being made possible by the flow cytometer, and during the course of that period of time, the invention of monoclonal antibodies. Later, I would be influenced by clinical investigators who had had a career of understanding basic science and applying basic science principles to the understanding of basic human pathological processes. So people like Hugh McDevitt, who really did a lot of basic work in the understanding of the mouse immune system, and yet applied that to human diseases such as multiple sclerosis or systemic lupus erythematosus, or indeed allergy, where understanding immunological systems and their susceptibility to disease was certainly benefited by an understanding of the basic genetics and the basic repertoire of cell surface proteins which determine the function of the immune system.
Speaking of the genetic code. I guess one of the biggest projects being contemplated now, more than that, it's on its way, is the Human Genome Project. What role do you see for cell sorters in that undertaking?
Well, flow cytometers and cell sorters are by their very nature able to provide information in a very new way. That new way is to make a multi-parametric instantaneous measurement possible. Thus, when a cell or a chromosome passes through the laser beam, it's possible to tell lots of different things about that particle at the same time. Thus, we might be able to determine its size, the number of active sites on the chromosome, for example, or to connect fluorescent probes to look at the number of copies of a given gene sequence in a chromosome. Flow cytometry, I believe, will play an important role in the cell sorter, and even more important role in elucidating the story of the human genome. The ability to create chromosome libraries is vitally dependent upon the ability to sort large chunks of DNA. Now, while this may be accomplished by electrophoretic means, the ability to relate back certain sequences in their structural relation to the rest of the chromosome is extremely important, because we know that large blocks of DNA may be shuttled around within the genome, and that this may be easily detected by means of fluorescence in situ hybridization, something which can be studied extremely accurately and extremely rapidly in low frequency on the cell sorter.
What motivated you to come from academia and a strictly clinical setting into a corporate industrial setting?
I came to Becton Dickinson because of the opportunity that this particular setting permitted me. The problem with being in academics and the problem with studying groups of patients in a geographically limited way is that there's a limited number of patients that have a given disease. When I was at the University of California, San Francisco, both as a faculty member and as a member of the Howard Hughes Medical Research Institute, I might have 15 or 20 patients, say, with a disease like lupus nephritis that I could study on a regular basis. Working as the corporate medical director and director for worldwide clinical research, I'm able to contact investigators around the world and study much larger collections of these patients than I could ever hope to accomplish as an individual investigator working in a single geographic site. So it was incredibly attractive to me to be able to do multi-center clinical studies. One of the advantages of working in the particular setting which Becton Dickinson has provided is the fact that we are interested in solving larger questions, and that we're not interested necessarily in studying very small groups of patients, but in using the flow cytometer to understand the immunological abnormalities which underlie specific disease states. And these disease states should be fairly universal. Thus, it's of interest to us to study patients in Sweden, say, in the United States, in the rest of Europe, in Latin America, in Japan, in Southeast Asia, and begin to understand the, if you will, immunological unities which underlie a given disease state, and then to also understand its geological or geographical, peculiarities. How is, for example, SLE different in Japan than it is in the United States? Is there an immunologic or immunogenetic component which underlies how the immune system may respond to a given etiologic agent or a virus? And these are the kind of studies which are only possible in a multi-center study, and certainly having command of the technology and the new tools as they are developed in a biological research setting and in an industrial development setting permits a scientist with an interest in clinical investigation to be there as the ideas arrive and apply them, those ideas, to new clinical settings as they emerge. So I look at the role that I provide as being a bridge between technology and clinical problems.
Good. Cut.
Brian, is this a more relaxed position? Do you like that better? Okay.
Okay, anytime, Bruce.
The medical significance of flow cytometry is multiple. We use flow cytometry.
Stop you just for a second. I still want you to talk to her.
Okay, sorry.
Yeah. Right.
And let's talk to you, too.
I've got to stop eating, I think.
I know, that's
That's why I don't wear jackets anymore.
okay, we're rolling. Let's start, Bruce.
What is the medical significance of flow cytometry? Well, I think the medical significance of flow cytometry comes anytime physicians need to measure cells to understand what's going on with a patient. We may use cell measurements to define a disease, the presence or absence of a disease, or whether we want to choose to use a particular therapeutic agent, or whether that therapeutic agent is actually working. Let's take a look at some examples. Medical applications of flow cytometry include HIV disease. We use the flow cytometer to understand how affected the immune system has been with the infection. Some patients are very fortunate in that they have a robust immune response to the HIV virus. Their CD8 cells may allow them to bring the virus into control. Some patients have a course where their CD4 cells or helper cells are depleted, and the flow cytometer allows us to measure the balance between the host response to the virus and the effect that the virus has on the host immune system. Another medical application of flow cytometry is in leukemia. In leukemia, it's extremely important to select the proper drug to treat a given leukemia. Leukemias may be grouped into three large categories. The first are so-called B-cell or lymphoid leukemias. The second might be T-cell, another form of lymphoid leukemia. And the third might be a myeloid type of leukemia. These leukemias need to be distinguished on the basis of their cell surface staining properties, and that allows us to select drugs which are going to be effective in eliminating the leukemia. Flow cytometry, because of its multiparametric nature, allows us to rapidly make the diagnosis, confirm the nature of the leukemia, and select the appropriate agent for its treatment. The last medical application of flow cytometry is in understanding cancer cells. In this country, breast cancer, colon cancer, and bladder cancer are significant public health problems. Flow cytometry allows us to study individual tumors so we can look at the heterogeneity, or if you will, the number of different kinds of cancer cells which are present. This allows us to select those patients which need intensive adjuvant chemotherapy from those patients which can benefit from a surgical cure or perhaps a surgical plus radiation cure for their disease. Thus, in all cases, flow cytometry allows us the opportunity to study cells, the locus of disease, to get precise, accurate, and reproducible answers, and to tailor our therapy to the patient's benefit.
Okay. 2 words or less, worldwide. This is a worldwide effort.
These medical studies are a worldwide effort. One of the things that we've learned at Becton Dickinson is that medical practice is different in geographical locations. The human population has great similarities, but also great differences. The needs in medical practice are different in different parts of the world. BDIS as a company is interested in making tools that are appropriate for medical practice in each of the world's medical markets. Patients are different worldwide. If one looks at problems such as malaria or infectious disease in Africa, if one looks at infectious disease in Japan versus the United States, there are different needs and different responses in patients around the world. Our technology is both generic and specific, and we try very hard to make sure that we understand the medical use of the answers which we provide so that we can provide the kind of information that's useful for physicians around the world.
Pride.
And in 1 minute, pride in the mission.
Oh, can you give me a little bit more than that? Pride in the mission? Why are we proud? What is it?
We're going to run out of tape.
Okay. We at Becton Dickinson are extremely proud of the contributions which we are making. Okay.
Look at it.
Okay. We at Becton Dickinson are proud of the contributions which we are making to medical science around the world. We feel a sense of mission in taking basic technology, putting it together in tools which work to solve problems of interest, both to the basic researcher, the clinical researcher, which is trying to translate the understanding of basic cell processes into disease processes, and to the practicing physician, who will take the information from flow cytometry systems and apply them to the solving of patient problems. If we can spare people unnecessary or inappropriate therapy, if we can find the right magic bullet for the right disease, then we at Becton Dickinson feel we have put tools that work into the hands of physicians who need them to take better care of patients.
All right, roll credits.
Good.
Okay.
What we're going to do now is try to show you exactly how a flow cytometer is used in the clinical setting. What we also want to show is the fact that these measurements are multiparametric and simultaneous. So we're looking at 5 or 6 dimensions of a cell at a single time, but we're restricted to doing that in 2 dimensions at a time. Kathy Grenier, who's a clinical research associate, and I will now show you a case of a human B-cell leukemia from a child to show you how clinicians would actually go about making a measurement to decide on what kind of leukemia is present and how is the best way to treat that leukemia. We're going to turn into the computer right now. What we have on this screen are 4 pictures of a childhood leukemia. We start up here and define the cells which we want the flow cytometer to examine. These cells are then looked at in 4 different windows. In this first window, we are Am I?
You were confused. When you do this, keep your hand up there until you're ready to move on to the next one.
Okay. Okay, you guys can see this. I can't, so just tell me what you want. Okay. I will walk through it. It's actually probably... This is an example. Oh, sorry. This is an example of a childhood leukemia. We're looking at the computer screen just as one would see in the laboratory. In the laboratory, a technician would be running the sample, and a pathologist or physician hematologist would be looking at the results. First, we identify the leukemic cells based on their size and light scatter properties. The next point is to move and look at the staining of those cells with monoclonal antibodies. In this first panel, we can see that there is very little nonspecific staining. Here is where the unique population of leukemic cells becomes apparent. This particular position of cell or cell cluster would never be seen in normals. It is a combination of an immaturity antigen with a marker of B cells. So already, just by this unique cluster of cells being present, we know this child has a leukemia. Now our task is to understand something about that leukemia. What about the distribution of proteins on its surface? Are there any other minor populations which we need to be concerned with? Kathy will now show us what the data looks like as it's acquired by the flow cytometer. You can see that the operator is reaching in and selecting the criteria for the signals which will be displayed. The machine is giving us a readout in terms of the details and the statistics associated with each of the quadrants. Here we're blowing up one of the populations of interest. We can identify the leukemic population and the residual normal cells in this individual. So what we've been able to do with this exercise is show how the flow cytometer, making 5 measurements at once, can allow us to identify the leukemic population accurately and also to see the residual normal cells so we can assess and understand the extent of disease. That's it. This diagram is a representation of the heart of a flow cytometer. We can see the light source which is shining on a cell. This cell is permitting, when in the light beam, 5 separate measurements to be made. We can see that the light is being scattered, which is a measurement of size. It's also being refracted at 90 degrees, which is a measure of the complexity of the particle or the number of small organelles within the cell. And we can see 2 antibodies, this one right here in orange and this one right here in green, which are giving fluorescence signals related to the presence or absence of proteins on the cell surface. So this is really the heart of what's going on in a flow cytometer. It's a cell passing through a light beam with multiple optical measurements being made simultaneously. This image represents the multi-parameter data which is being acquired from a flow cytometer. This particular sample was taken from an individual who's infected with the human immunodeficiency virus. Let's look at this image. Shown in the color are the various cell groups which make up human peripheral blood. The green are the lymphocytes, the red are monocytes, and the blue are neutrophils. The power of the flow cytometer is the fact that we can make multiple correlated measurements. So in this case now we see the neutrophils, here we see the monocytes, and here the lymphocytes. All of these panels represent the cells displayed in different parameters. So in this case we're looking at size versus the display of a certain antigen. These pictorial representations show the power of flow cytometry in its ability to see the multiple dimension of a cell's properties. Did I goof you up? Sorry. Let me do it again. This image represents the white blood cells obtained from an individual who has been infected with the human immunodeficiency virus. And it shows how a flow cytometer can make and represent multiple bits of information in a simultaneous pass through the flow cytometer. Shown in this image are the three major classes of white blood cells. The lymphocytes, shown in green, the monocytes, shown in red, and the neutrophils, shown in blue. The ability to identify cells by color and by parameter is extremely important. These measurements are just on the basis of physical property, size, and complexity. These measurements are the same cells now displayed in terms of their fluorescence characteristics. So blue are the neutrophils, green are the lymphocytes, and red are the monocytes. Notice how each of the different cell lineages or types has a unique address or space in each of the graphs. One can move and put combinations of physical properties, in this case size, versus fluorescence property. So this panel of 6 images represents the multiple and multi-parametric measurements which are possible with a flow cytometer. And each image tells us something unique about the cells being studied. This slide represents the difference between a normal individual and an individual who's infected with human immunodeficiency virus with regard to the activation set of the different cells which make up the immune system. In the normal, we can see the normal B cells or antibody-secreting cells which are identified by their fluorescence here. T cells are identified by their fluorescence with this marker which is associated with T cells. Notice that in normals there's a small number of activated T cells. They're present, but not in great number. If we now turn to an individual infected with human immunodeficiency virus, we see a decrease in the number of B cells, some activated non-B cells, and a large increase in the activated T cell population. This slide represents how the flow cytometer can take a single marker, which can be both an activation marker and a cell identification marker, and show how it is an appropriate way of studying a disease state. This example shows how we can read the signature of the immune system by making multiple two-color immunofluorescence measurements. Shown in panel A is the range of normal display of activation antigens within the so-called cytotoxic suppressor compartment. Notice that they're tightly clustered near the bottom of both these activation antigens in this direction and in this direction. Moving to the next image, we see individuals who've been infected with various viruses but not human immunodeficiency virus are pretty much similar to the control group. However, after an individual has been infected with human immunodeficiency virus, or HIV, we can now see a spreading out of the immune response. Each of these triangles, or diamonds rather, represents an individual patient which has a certain value for an activation antigen. Notice as the disease progresses into more advanced clinical stages, here the lymphadenopathy phase, how patients can now be identified by a clustering of values. This suggests discrete states within the immune system which have been reached. As the disease continues to progress, more activation is seen and more dispersion in the state of the immune system can be seen. Finally, in frank AIDS, one can see that very, very high states of activation exist in these patients. Thus, we can use the flow cytometer to create a footprint or a map of the immune response to HIV. Flow cytometers can also be used to study cancer. We understand that not all cancer cells are the same, and part of the process of developing a tumor involves acquiring new genetic information, or if you will, shuffling the genetic information or chromosomes which are present in the tumor cell. We can see here a hypothetical diagram of how cancer cells proliferate. The colored nodes indicate changes in DNA content. This DNA content can be identified, studied, and quantitated by the flow cytometer. This slide represents how actually measurement of DNA content is possible. The flow cytometer can recognize the propydium iodide molecule by its fluorescence when it binds to DNA. This binding is done in a quantitative fashion so that there's a fixed relationship between the amount of propydium iodide and the amount of DNA. This is manifest as a fluorescence emission. By studying the height, width, and amount of signal, we get a direct measure of how much DNA is present within an individual cell which makes up a tumor. This image represents an actual measurement of DNA content in a cell which is rapidly dividing. By the position, we are able to establish each of the compartments or activities of the cell. This group represents cells which are not dividing and are quiescent. This group represents cells which are increasing their DNA. And this group represents cells which are about ready to divide into two daughter cells. This is an example of what one would see if one was doing DNA analysis sitting at the flow cytometer. This slide represents the kinds of measurements that are possible using a flow cytometer to study cancer or proliferating cells. We are trying to put cells into one of the three categories which I just identified. Here we see quiescent cells which have not entered a period of DNA synthesis. Here we see cells which are in the process of DNA synthesis. And here we see cells which have completed the process of DNA synthesis and are ready to divide. By measuring cells in the S phase or the G2M phase, it is possible to make decisions as to the most appropriate course of chemotherapy to give for a given tumor.
This afternoon, we're in the production facilities of Becton Dickinson Cytoimmunocytometry Systems Company, talking with our tour guide, Richard Owens, who is Director of Instrument Operations at BDIS. Richard, could you tell us what happens in this room?
Yes, certainly, Ray. Here we're going to be showing the testing process of our FacStar Plus product. When the unit is brought into the lab, we in fact install a laser onto the back panel of the actual machine. The alignment of the laser is then carried out through prisms and lenses in this part of the machine to align the laser beam in the main cell path, which is in the center of the machine. The adjustments are made.
You can go right in and point to us.
Okay. Set into the middle of the machine in this aperture here. We adjust the mechanics such that the laser beam is aligned for our forward scatter measurements and for our fluorescent channel side scatter measurements. Once that setting has been achieved, we then position and adjust the drive nozzle such that the droplets that are then passed down through the laser beam can be positioned and the elements collected in the catcher tubes at the base of the cell system. The gating arrangement for the drive is controlled through the computer and electronic console modules, where we have the ability to set the gating parameters relevant to the flow of the droplets in the machine. This enables us to take the electronic signals that are given from the fluorescent housing system, which are characteristic of the fluorescence of the sample. We can set thresholds such that we can in fact select individual cells and collect them separately for the researcher to take to another part of the operation to do further experiments with the particular sample that he has got. So the electronics console is the mechanism that we use for setting those particular parameters. The whole process in this room is purely based upon the final alignment of the laser and the droplet stream and calibration of the electronics for the performance of the machine.
Good, thank you.
Just you step back and if you just get a cover shot of the FacStar Plus, right, just a clean. Right, actually, just stop there for a second. We're still rolling, right? Good. And now pull out the cover shot of the instrument. Just go ahead and pull out. Good. Okay, and just hold there. Okay, excellent.
Pause. Rolling. Hold for speed. Speed.
Okay, we have now reached the system test lab at the side of the manufacturing area. For this particular filming, we've actually opened the doors in order that we can see the lab in its entirety. Normally these doors would be shut as a result of having lasers in operation as a safety precaution, and the light levels would obviously be somewhat reduced as well. But what you see happening here is some typical activity that would normally go on behind closed doors in actually calibrating and aligning the FacStar Plus instruments.
Keep rolling, please. Can we ask him to move out or in?
Yeah,
Okay.
he's gone.
Okay, good. Just keep rolling for a second. Okay, that's fine.
We have now moved on to workstation 8, having moved past station 6, which is where we will have put on the beam focus assemblies onto the main casting. Our workstation 7 is in fact the optics room, which we saw earlier in our process. It's just an operational numbering sequence that we've adopted for the control of the material in our manufacturing area. At this particular workstation, the operator is building the head adjust assembly, which is used to control the nozzle drive in the final machine to locate the stream in laser beam in order that it not only intersects the laser beam but also then fires into the collection system in the catcher tray at the bottom of the assembly of the FactStar unit.
Rolling. Hold for speed. Speed.
We've now arrived at the end of the manufacturing assembly line, which is in fact our workstation 9, and here you can actually see a base casting now with all the elements assembled onto it, which is shortly to go off to our test process to start the final alignment and testing of the product. At the left-hand side of this particular assembly, we can see in the upper portion of the picture is in fact the preamplifier printed circuit board, which is used for taking the signals from the photomultipliers back to the electronic console. In the lower portion is in fact the back of the fluidics assembly that we saw being assembled earlier in the production process. On the right and to the top of the assembly, we can see the fluorescence channel block, which contains the 4 photomultipliers that are used to collect the fluorescent signals from the samples when they've been excited by the laser as the droplets pass through the laser beam. This is now the completed system and is the end, in fact, of our manufacturing of the assembly process.
All right, stop tape.